Okay, so we last time thinking about Woese and Fox's phylogenetic tree of life. Remember, they sequenced ribosomal RNA and came up with three basic kinds of ribosomal RNA, and so they assigned organisms into three different domains. And the advantage here is this gives us evolutionary relationships. Okay, so more and more today, when we classify organisms, we are interested in those phylogenetic relationships. I wanted to take a minute as we're kind of switching into chapter 11 and thinking about some of these characteristics as how they pertain to taxonomy and classification. We finished, I think, actually last time on this slide where we were comparing some characteristics of the domain bacteria and the domain archaea. And really, a lot of these differences I'll try to highlight a little bit when we go through these actual structures. So it's difficult to kind of pick apart these structural differences when we haven't really talked about those structures yet. Today we're going to talk a lot about peptidoglycan. So this is a big difference. Bacteria always have peptidoglycan in their cell walls. If they have cell walls, almost all of them do, where archaea never have peptidoglycan. They have structure that's similar to that, but it's not the same. And of course, if eukaryotes have a cell wall, like fungi or plants, they never have peptidoglycan in them either. So that's kind of the main difference. There's again, lots of other structural differences. We don't go too in depth about membrane structure, but membrane lipids are different in bacteria and archaea, and even different in the eukarya as well. We see the archaea have a lot of similarities in metabolism with bacteria, but then they seem to share genetic similarities with eukaryotes. So in terms of, like, just an example, they use the start codon AUG that encodes for methionine, just like eukaryotes, and bacteria have a slightly altered first amino acid there in formal methionine. So just an example. We'll be trying to kind of bring up differences kind of as we go. But we're going to kind of just do a little part of chapter 11. Chapter 11 is a survey of prokaryotes, so it kind of attempts to go through, all the different genera or big groups of prokaryotes. So we get into a lot of environmental organisms, and we're not going to worry about a lot of that, but it does go through some basic things that we use to classify bacteria. So we need to hit some of that terminology. And one of the first things that we think of is the shape of cells or morphology. So terminology you should be aware of. Round cells we refer to as a coccus, or plural cocci, and rod-shaped bacteria we call bacillus, a bacillus, or again the plural bacilli. Okay, so those are by far the most common morphologies that we see. If you get something that's halfway between a coccus and a bacillus, then we call that a coccobacillus. Okay, somebody told me that was an MCAT question once. What shape is a coccobacillus? Okay, if you are in lab, you are only going to see cocci and bacilli. But I want you to be aware that there are so many different shapes out there. There's comma-shaped bacteria that we refer to as Vibrio. There's our helical bacteria, our spirillum and spirochetes. What's the difference between those two? I mean, they do look different here, but they both move in a corkscrew like motion, which is kind of cool to see. The spirillum are a little bit more rigid, and they have external flagella You can't see the flagella. We're going to talk more about flagella today. You can't see the flagella on this picture, but they usually have tufts of flagella at one or both ends. And the spirochetes are a little bit more flexible. And they have this kind of bizarre flagella that's actually considered internal because it's covered in a membrane that goes down the whole length of its body. Okay, so both of them, again, move in corkscrew manners. Again, little bit of difference between them. I'll give you a few more examples in the next slide. But there are some organisms that really don't have any defined shape, and we call those pleomorphic. Sometimes they have a different shape depending on the environment they're in. Sometimes they don't have a well-defined shape just because they don't have a cell wall. Okay, again, there's one genera of bacteria that doesn't have a cell wall. And then especially in the archaebacteria, we see a lot of other really odd, bizarre shapes, like star-shaped, or there's some that are actually square. So there's a lot of kind of crazy things out there. Just, again, the difference between the helical bacteria, and check out this link if you get a chance. It's just like a minute long. It's showing the motion of some spirochetes. Just write that down here. Again, looks like some dark field microscopy. Remember, that's a big advantage of dark field. We can view cells when they're still alive. We don't have to stain them. And you can just kind of see them buzzing around a little bit. That's kind of fun. Remember, here's some spirillum. Here, the brown things here are the flagella. So it's kind of hard to tell what they are. They're typically out of one end or the other. Again, you can kind of see this one is more there. Again, a little bit more rigid. The spirochetes are a little bit more bendy when they move. This organism, this spirillum, is Helicobacter pylori. That's an example, again, of a spirillum. That's an organism that causes gastric ulcers. And an example of a spirochete, I believe this micrograph is of Borrelia burgdorferi, which is the causative agent of Lyme disease. All right, so check out that video if you get the chance. All right, another very common way that we classify organisms is how they arrange their cells. Okay, so here's some examples of cell arrangements for round organism. And how they arrange is going to depend on a couple of factors. It depends on the plane in which the cell divides. And you can see here, for example, this cell, it only divides in one plane. Okay, down here at the bottom, we've got a cell that's actually able to divide in many different planes. Okay, so that makes a difference as to what arrangement we see, as well as the separation of the daughter cells. Okay, so in this case, this is labeled in your book, but we'll write them in here. The diplococci, whoops, didn't spell that right. Okay, pairs of cells, so they divide in one plane, and then usually only two of them stayed stuck together. Or if more of them stayed stuck together, then you might see some long chains. If it divides in two planes, and then again a little bit stays stuck together, these are described as tetrads, groups of four. And if it forms more of a cube shape, then we call those, let me write that a little bit more neatly so you can read my writing. We call those sarcina. Okay, in other words, cubes. So packets of 8. In reality, when you look at these under a microscope, it's sometimes really hard to tell the difference because it's hard to see 3D, whether there's 4 there or whether there's 8. But again, we saw that cell arrangement. That would give us a hint as to what organism we see. And then if we see clusters or big groups of cells, that's when the cells stay stuck together and it divides in many different planes. Okay, so we see a good variety of the cocci, different cell arrangements, not quite as much variety when bacilli are dividing, because bacilli, right, they get longer and longer and longer, and then they divide in the middle, okay? They never get fatter and fatter and fatter and divide this way. Okay, so you don't see that. So that limits the possible arrangements here. If you've got a pair, we just call those diplobacilli. If they're chaining, this prefix strepto means chaining. We can get streptococcus as well, and that's just another way to mean chains of round cells. And then sometimes we get this formation that's referred to as palisades. And the way that happens is sometimes referred to as a snapping division, where the daughter cells just still stay attached by just a little bit there, and then it kind of hinges, and we get the cells kind of stacking up on themselves. Okay, so that term palisades, if you were to look at like a fort back from the 1800s, and their fence would be, logs lined up like this, that was referred to as palisades. So that's where this cell arrangement name came from. All right, some other things that are typically used to categorize cells. We're going to talk more about endospores in a little bit more depth, but just to mention here, the formation of endospores is a survival mechanism. It's not reproductive in bacteria. It is in fungi, but not in bacteria. And there's only a few genera of bacteria that are able to sporulate and make endospores. So if we do a special stain and we can see the spores, which are these little green ones, or here's one where the spore is still inside the mother or vegetative cell, then that gives us a big hint as to, narrows down a lot as to which, classification the organisms belong to. So we'll talk a little bit more about how that staining happens and that sporulation cycle, but that's good for our purposes now. So again, some simple criteria that's used for classification. We talked about morphology. We talked about cell arrangement. We're going to talk in more detail about gram staining and gram reaction. So whether an organism is gram-positive or gram-negative is generally the first question we ask when we're trying to identify an organism. So that's a really important thing for classification. Let's see whether it makes endospores or not. And lastly, what's the colony morphology look like? When you get good at recognizing colony morphology, you can look at a colony and say, oh yeah, there's some whatever cells growing there. Remember, colony is what we can see with our naked eye. So here we've got colonies growing on a plate. What you see through the microscope, you're seeing cells through the microscope. So just remember the distinction there. And when you first start looking at bacteria, you might say, oh yeah, it's just a bunch of white dots. That's what colonies look like. Well, when you get a little bit more practiced at it, you can, again, start to notice a distinction between different colonies. And I didn't post this because I didn't want you to be responsible for all this terminology here. Just be aware that there's many different shapes of actual colonies. They're not all just perfectly circular. They can be, irregular, or, spindly like this, the margin can be way different. Sometimes the colonies are raised up, sometimes they're flat. Of course, the size can be different. They can be dry or shiny. Of course, there's lots of different colors they can be. So lots of different ways that we can describe those colonies, and again, might give us a hint as to what organism we're dealing with. Kind of a level above things we can tell just by seeing our biochemical tests. And we're kind of bopping back to chapter 4 here, just briefly here. But there's lots of differential tests that we can use in the lab to identify organisms. So remember, we talked about differential media. That's media that has some ingredient in it that causes certain organisms to have a different appearance than others. All right, so for example, here's some sugar. They're probably, in lab, we call them sugar fermentation tubes, where we put different sugars in different tubes and then add our bacteria and see if they can metabolize those sugars. And if they do, then we get a nice color change. And what color it is depends on what pH indicator you put in there. But again, we can test for what kind of sugars an organism uses. Does it make a gas, for example, when it uses that sugar? That depends on what kind of fermentation pathway the organism has. So here, there's actually a little inverted tube inside here, and here's a gas bubble here in this one, which is different than whatever this organism is. So that's just one more way to characterize it. Here's another test for a particular gas, hydrogen sulfide. Here's this one's positive, this one's negative. Just as an example, we have lots of tests that we do for particular enzymes. More rarely, we might use antibiotic sensitivity. I guess we don't often use that to actually identify an organism, but it can help us characterize it, and that's something that if you're in lab, you'll do that. I did mention in lecture a couple of other even more complex criteria that we would use for classifying, just because it was on my old slides that I used, that I meant to leave out this time. But we already talked about that ribosomal RNA sequencing, how, again, we're moving towards more advanced criteria than that, or some kind of serological analysis like using antibodies to identify organisms. So we're moving a little bit more in that direction. But if you want to look up, kind of a consensus of experts on taxonomy, you would want to look up it's more like a tome. It's more like I think it's 5 different volumes of Burgess' manual, and basically it has classifications based on evolutionary relationships as well as biochemical tests, and anything you wanted to know about any organism, basically you should be able to find it in Burgess' manual. All right, so we're going to press forward into chapter 3, and this is a chapter that we cover pretty extensively. So I'm hoping to get out a little, hey, this is a tour of the Mastering Microbiology website, just so you're aware of some of the study helps that are there if you haven't looked around that much. But they have a lot of practice questions, and chapter 3 is a great one to kind of explore that a little bit, because pretty much covering almost everything that's in the book in chapter 3. So we're going to try to highlight differences between prokaryotic and eukaryotic cells. We're going to run through the prokaryotic structures first, for the most part, and then go through eukaryotic structures a little bit more quickly, paying particular attention to cell walls. We'll look at cell membranes and flagella, again, as well as some of those organelles. Okay, so this is really the framework for what we're, a lot of structures that we're going to be talking about all throughout the semester. So it's kind of, fundamental things. All right, so review. What's the difference between prokaryotic and eukaryotic cells? Of course, the prokaryotes, this are the domains bacteria and archaea. And what they have in common is that they have no membrane-bound nucleus. Okay, that should be review. Every once in a while, I'll have a student tell me, oh, the difference between prokaryotic and eukaryotic means prokaryotes have a cell wall and eukaryotics don't. Remember, that's the difference between plant and animal cells, so try not to get goofed up there. Prokaryotes also are going to lack internal structures bound by a membrane. Okay, so not only they don't have a nucleus, but they don't have all the organelles that eukaryotes do as well. Typically, they're going to be also a lot smaller. Typically, they're going to be about one micron or micrometer in diameter or less. Okay, so here's an example of some bacterial cells. See the units here, about 1 micron. This it looks like it's probably a paramecium. Okay, much different units here, so way bigger. All right, so just to be complete here then, eukaryotes, basically all the opposites. They have a nucleus and membrane-bound organelles. Typically at least 10 times bigger, 10 to 100 micrometers in diameter. And some examples of eukaryotes that we're going to talk, we're going to mention algae at least. We're going to spend a little time on microscopic fungi, of course, plants and animals. We don't really get into in this class. We will talk a little bit about protozoa. All right, so those are all, we'll get to those. What have we left out? Well, we haven't talked about viruses yet because, of course, viruses aren't really considered prokaryotes or eukaryotes. They're considered acellular. They don't really have a cellular structure, so we will get to those as well. All right, so looking on the outside and working our way in to a bacterial cell. Don't worry, we're spending a lot of time on the structures on the outside because there's not a lot that's actually inside a bacterial cell. The glycocalyx, this is a general term for a slimy substance secreted outside of the cell. Okay, and most bacteria are going to produce some kind of glycocalyx because it's, when we look at the functions, it's just so advantageous to the cell energetically to do it. What's the composition? Most of the time, polysaccharide. Okay, a sugar composition. There are exceptions there. There are some bacteria that have more of a protein composition to their glycocalyx, but commonly, again, carbohydrates. Okay, so function of glycocalyx. The big three here, resistance to phagocytosis. And I'm kind of assuming you know what phagocytosis is. We're going to talk about that in a little bit more detail probably next time. But if you've got a bacteria cell that's trying to infect our body, our non-specific immunity, our white blood cells like our macrophages and our neutrophils have a lot harder time. Actually, neutrophils aren't typically phagocytic. I don't know why I said that. Macrophages are going to have a lot harder time engulfing a bacterial cell if they're covered in some kind of glycocalyx. Okay, so it's a protective feature there. Also good resistance to drying out, desiccation, right? You've got a bacteria that's coughed out into the environment from our lungs. It's going to stay viable for longer on that desktop, for example, if it has a glycocalyx surrounding it to keep it moist. And another big one is attachment or adherence to some kind of host structure. For example, you have lots of bacteria that form a glycocalyx in your mouth in order to adhere to your teeth. Okay, so that keeps them in the mouth instead of having them be washed down into the digestive tract. Okay, so also adherence and to other bacteria in the formation of biofilms. Okay. So big mats of bacteria. All right, a glycocalyx can be considered either a true capsule or a slime layer. And keep in mind that this is kind of a continuum. Sometimes it's kind of hard to tell, well, would you consider it a capsule or a slime layer? Basically, a capsule is more basically tightly bound to the cell wall, maybe a little bit thicker, a little bit more rigid, where the slime layer is going to be just a little bit more loosely bound. Okay, so they both have the same function, the same composition, but a true capsule is going to do all those functions a little bit better than a slime layer. So you can see here a true capsule, that's the orange here. The slime layer, it's just a little bit more loosely organized, maybe around the cell. Okay. Okay, so remember, most bacteria make some kind of a capsule. If you are in lab, you will be doing a capsule stain here in a couple weeks, and so I won't go through that in real detail, but I can see my labels here didn't come up really good, but so here's the bacteria. The staining procedure that we do looks a little bit different than this, but if you can stain the bacterial cells and you stain the background, then basically the capsule part stays unstained. So you have this kind of big halo around the cells, and like I said, the only way to really visualize this is to do this, stain the background so that you can see those, because it's very difficult, if not impossible, to actually stain the capsule itself because it's not charged. So again, I'll let your lab professor talk more about that, but that's typical capsule stain there. Okay, so as we are moving into the cell, the next structure that we may come in contact with is the flagella. And only some bacteria are going to have flagella. Cockeye-shaped or round bacteria, as far as I know, don't have flagella. There may be some exception there, but this is mostly going to be the other morphologies that we see, like bacilli and Vibrio and spirochetes. Okay. Of course, the function of the flagella is to have that bacterium move around in a purposeful way. They usually are going to move towards some attractant. So it helps the organism perform chemotaxis or maybe phototaxis if it's moving towards a light, if it's a photosynthetic bacteria. All right, so if we look at the structure, and we'll see the diagram, the graphic on the next page here, but there's three parts, the filaments, okay, and that's made of a single protein, not too hard to remember, flagellin, 1G. 2 L's. There we go. Okay, and there's, if you've ever, just as a little aside here, if you've ever heard of the organism E. coli O157H7. This is sometimes we get numbers like this in association with bacterial names, and this is a particular strain of E. coli. It's the really nasty, deadly one, the one that causes food poisoning, that can cause patients' kidneys to shut down. What all these little numbers mean back here, well, the H is actually the different variations of flagellin. And it's H because it stands for a German word, but we have at least 7, I think there's more than that, different variations of that flagellin protein. So we can use antibodies to kind of figure out which variation that is. The O, these are some cell wall antigens, but again, just sort of interesting. There are different variations of that protein that make up the flagella filament. There's also going to be a hook region, and then the basal body. That's the region that anchors into the cell wall. Okay, so most of the time, there is an exception there. It's not enclosed in the cell membrane or any kind of membrane. Okay, but that's bacteria. Okay, there are we'll look at flagella in eukaryotes as well, and there's a ton of differences between bacterial flagella and eukaryotic flagella, so stay tuned for that. So here's, again, the filament. So this is going off like this of the flagella, the hook region, and then this is the, what we would, these rings is the basal body, so where it's anchored into the cell wall, and it looks like even into the membrane here as well. So it's just the two different diagrams. This is supposed to be a gram-positive cell wall, and this is a gram-negative. Again, we haven't talked about the difference between those two yet, so don't worry about that too much, but there's just, it's a little bit more complicated, apparently, anchoring into a gram-negative cell wall. And notice here, these arrows up here, the bacteria flagella are always going to be spinning. Okay, so they're always rotating. So we'll look at that a little bit more closely when we look at their motion. I want to talk first about the arrangements of the flagella. I've got some terminology here that hopefully will make sense and won't be too much of a burden. Paratrichous arrangement. That just means that it's basically all over the cell. So here's an example of paratrichous. You can see there's flagella coming out everywhere around the cell. And this is actually very common for bacteria to have paratrichous flagella. E. coli, for example, has paratrichous flagella. When we see that organism in lab, it's easy to forget that, because when we are doing our kind of regular staining procedures, we can't see the flagella. Okay, you have to do a special flagella stain to be able to see them with a light microscope, and apparently it's fairly difficult to get that to turn out right. So peri means around. So if you look at the perimeter of a room, you're measuring the distance all the way around the room. So hopefully that helps you remember that. Monotrichous flagella, that just means there's one flagella. So they don't use that particular term in your book, but here's their picture of a single flagellum. Polar just means that it's one end. You would never have a flagella like sticking out just like that, just all by itself. You would always have it at one end or the other if it only had one. Okay. Lophotrichous means you have a tuft of flagella at one or both ends. Okay, so again, they don't use this term lophotrichous. Apparently lopho means tuft. They just call it a tuft in the book, but you can see a kind of a bunch of flagella sticking out from one end. And then lastly, amphitrachus. That means that there is a flagella, one or usually just one at both ends. Okay, so amphi, that prefix means both. If you have an amphibian, that's an organism that lives in both the water and the land. Okay. And then the last one is on the next page, kind of flagellar arrangements, is that axial filament that we mentioned before with the spirochetes. Okay, so remember this is that particular morphology. Okay, that's those long, one of the classes of those helical bacteria. And it is, like I mentioned before, covered in an outer membrane and runs down the whole length of the cell. So here's the axial filament here. And actually, if you look at a cross section, here's the axial filament. There's also actually all these little endoflagella in there that actually spin around and rotate, that makes the organism, again, move in this corkscrew-like manner. And this is a picture of an example of another spirochete. It's Treponema pallidum. That's the causative agent of syphilis. Okay, so it helps the organism move through more viscous material, more gel-like material when it kind of moves in that corkscrew type manner. All right, let's look a little bit more closely at flagellar motion. This is the graphic from your book. Basically, the motion is going to depend on somewhat by the arrangement. So if you have a monotrichous flagella, you just have one flagella at one end. If you change the direction of rotation, then basically you change the direction of motion. But if you've got a paratrichous flagella like this organism here, basically if the flagella are spinning in one direction, and that's counterclockwise, and I always tend to forget which way is which, so that's probably not something I would ask you. If they're moving counterclockwise, and it's kind of hard to see, yes, this is counterclockwise here, then the flagella all work in concert and together and produce a very, basically, motion in a straight line. Okay, if, though, whatever's attracting them, they start to realize they're not going towards the attractant, they need to change their direction. And so all the flagella, if they start spinning clockwise, like they are here, then that produces a tumble. It's written over here. Okay, so basically the bacteria will kind of spin around in a lot of different directions, and when it changes back to the other way, it will move in a different way. So as it moves towards this attractant, it might move, in a really haphazard-like way till it actually gets there, okay? Because the tumbles, it's just kind of guessing and going out into a different direction. So I did post a couple of links that shows this really nicely. This one is just a really short kind of animation that's kind of fun. This is a little bit longer. This one's more like 10 minutes, but if you look around the time of about 1:15, that's kind of the best view of purposeful motion where you get runs and tumbles. You have to kind of look really closely at cells to see that, but it's kind of fun to watch. All right, a couple of similar structures, at least they look similar, that you may encounter on gram-negative organisms. I'm going to get to that cell wall and talk about what that means to be gram-negative shortly. Are fimbriae and pili. And these are two structures that actually are both, I'll just write this in the middle, have the same composition. So they're composed of a protein called pilin. Okay, so because they both have the same composition, sometimes these, whether it's called fimbriae or pili, some sources say, well, it's really the same thing. Typically, though, we call this structure fimbriae if there's lots of them all around the cell. Okay, so it's these kind of shorter hair-like ones. This longer one, like right here, this is a flagella. Okay, so the fimbriae are the shorter ones, and lots of them. And typically, if we call it a pilus, that's a singular, or pili, typically a cell only has maybe one or two. And these are typically a little bit longer than fimbriae. Okay. The main function of fimbriae is for attachment. So E. coli, for example, has lots of fimbriae that it uses to attach to the epithelial cells of our large intestine. If it didn't have that attachment structure, then it would just get washed out of our digestive tract. Pili, the function here is in conjugation. We're going to talk more about conjugation later, but that's a kind of horizontal gene transfer where one cell extends a pilus to another cell, and that pilus actually draws the cells closer together, like this, and then a pore opens up, and the one cell shares DNA with the other cell. Okay, so that's a little overview of what conjugation is. The DNA is not actually traveling through that pilus. We used to think that was the case, but it just, a pore opens up in between them. So, again, mostly in terms to transfer DNA, sometimes that motion of bringing the two cells together is considered motility, kind of a twitching motility. Of course, it's fairly slow. It's not like the runs and tumbles we see with other kinds of flagellar motion. Okay, and again, there may be an exception there, but for the most part, we just see fimbriae and pili in gram-negative organisms. All right, we're going to spend a good bit of time here on the bacterial cell wall. This is a structure that we will kind of keep returning to throughout the semester, especially when we talk about antibiotics, because our biggest class of antibiotics are ones that target the bacterial cell wall. The function, well, it does determine the shape of the cell or the morphology of a cell, and it imparts a lot of strength to that cell surface. Okay, so it enables the cell to withstand changes in solute concentration. If you think about it, an animal cell, and we will review osmosis, probably we'll get to that very next lecture. Animal cells are very easily burst if you put them in a low solute concentration, where bacteria cells, because they have this nice strong cell wall, can withstand a lot more influx of water, as long as that cell wall is intact. So, composition. All bacteria cells are made of peptidoglycan, or they have peptidoglycan in them. And the amount of peptidoglycan they have can certainly be different. But this molecule, if you break this up a little bit, you see that glycan, and that should make you think sugars. Okay, so alternating sugars, and those two sugars are N-acetylglucosamine we abbreviate that NAG, and N-acetylmuramic acid. Okay, NAM. Oh, getting tired. I can't write right here. Okay, NAM. All right, so we have, alternating sugars here. Oops. Again, I'm having trouble with my brain here. Here's the link in between those two. I didn't draw that very good. So, again, NAM and NAG alternating. You'll see that in the next graphic. Okay. The other portion here, peptido, that should make you think peptide. So that peptide, that means there are some amino acid cross bridges, short segments of amino acids. Okay, and just remember that archaea, eukaryotes, if they have cell walls, they never have peptidoglycan in it. Okay, so I'll just peptidoglycan, PG. Okay. I want to talk a little bit. Here's this is from your book here. If you're interested, the structure of NAG and NAM, you'll see is very similar to glucose. And let's see. So the color coding here, I guess the gray NAM and NAG are the same here at this position, and the brown here is the NAG is similar to glucose there, and NAM is different. And you don't have to know the structure, just the point is that, hey, they're not that far away from the simple sugar glucose. Okay, and here's your book's graphic of what that peptidoglycan, what we think it looks like. So alternating NAM, NAG, and then you have cross bridges from one layer to the next, up and down, and also cross bridges this way from side to side. So the analysis that I've heard that makes sense is that peptidoglycan is kind of like a chain-linked fence. It's pretty rigid, pretty solid structure. Okay, I want to go a little bit beyond. Your book doesn't talk at all about cell growth, but I want to mention a couple important aspects of how that cell wall grows, because this does come into play later on when we talk about how our antibiotics work. So the thing to remember is that, right, as a cell gets bigger and bigger, it gets bigger and bigger, and then it divides. It has to be expanding its cell wall. And it has to do that kind of carefully because it needs to maintain that protection from changes in osmotic pressure, basically. Okay, so there's a couple of enzymes that are important here. Let me write in cell. Oh, I already did write that part. Okay, never mind. Okay, so cell has to add more peptidoglycan units, so basically has to add in some NAM and NAG subunits in order to grow. And those are synthesized inside the cell and then are transported out to the cell wall. Okay, so again, some enzymes, bacterial enzymes called autolysins. Break those glycosidic bonds between the two sugars. So let me try to draw this again here a little bit better than my attempt before. But here's the glycosidic linkages, and we've got the autolysins that are going to be cutting that, as well as those amino acid cross bridges. So, you can think about that has to be done kind of carefully and regulated to kind of keep up with the bonds that are going to seal those linkages back up again. So if you have lots of cutting taking place and not enough sealing, then that's not going to keep the cell intact. Okay, other enzymes, transpeptidases, they're going to reseal the bonds of those amino acid cross bridges after new sugar subunits are added. Okay, so I'll come back to this in a minute, but this was the best graphic I could find of this. Okay, so here's inside of the cell, and here's the cell wall on the outside, right? Those NAMs and NAGs are synthesized in here, and then they're shuttled through a particular kind of pathway or channel in the membrane, bactoprenol, which we won't worry about that here, but then the new subunits again are after the autolyses have cut these bonds, the new ones are put in, and this TP, that stands for the transpeptidase, or a transpeptidase enzyme that seals up those linkages again. So, we'll talk about this more later, but penicillin actually binds to transpeptidase and basically keeps it from catalyzing that reaction of sealing up those amino acid cross bridges. So basically what you get is a loss of the integrity of the cell wall. And if you look back at this kind of short time-lapse video, it shows some cells bursting when they're treated with penicillin due to influx of water. Okay, so again, the cell has to grow, everything has to be very carefully regulated in order to keep that cell wall intact. Okay, all right, so let's talk about the difference between gram-positive and gram-negative cell walls. Okay, so a gram-positive cell wall, I should mention, sometimes students get the misconception that gram-positive cells have a certain shape and gram-negative cells have a different shape, and the Gram reaction doesn't have anything to do with the cell morphology. There's gram-negative cocci, there's gram-positive cocci, there's gram-negative bacilli, there's gram-positive bacilli, for example. Okay, so gram-positive, so a couple hallmarks of these cells: they have a very thick layer of peptidoglycan. And I'd say they're a little bit less complex than gram-negatives. Okay, so a very thick layer of peptidoglycan. I was just rechecking how much peptidoglycan it has, and the number that I came up with is somewhere between 40 and 80% of the dry weight of the cell wall is peptidoglycan. Now, in my old notes, I had that it was 90%. So usually a majority of the cell wall, we'll say, is peptidoglycan. Okay, and so I'm not going to go through the gram staining procedure real carefully. If you're not in lab, just so you get an idea of this, if you're in lab, you're going to do it lots of times. But the primary stain is a purple dye. It's called crystal violet. And then we go through a few other steps, like adding some iodine that really pounds that crystal violet in there. We do colorize for a few seconds. And then gram-positives, because there's so much peptidoglycan in the cell, they hang on to that purple dye really well. And even when we add a counter stain or our secondary stain, they still hold on to this purple dye. So basically they stain purple when we gram stain. That's a reasonable thing to know. Again, better to kind of think through that rather than just memorize it, but the gram negatives get decolorized really easy, and they take up the color of this secondary stain, which is red. Just write that there. Okay, and the way that I remember this, so gram positive, thick cell wall, lots of peptidoglycan. So lots of peptidoglycan. Gram negatives, all right, thin peptidoglycan layer. All right, so that's one way that can help you remember that. And if they have lots of peptidoglycan, again, they hold on to this purple dye much easier. Okay, the other thing that they have in their cell walls, gram positives, that gram negatives don't, is that they also have something called teichoic acids and lipoteichoic acids. Okay, and they just help with the cell shape to some extent. They play some role in regulating cell division. They anchor the cell wall to the cell membrane. Okay, so again, you don't find these in gram negative cell walls. Oh, I guess I just gave myself a little extra room there. All right, so here's your graphic from your book, and we labeled this graphic in class. This pinkish portion here, that's the peptidoglycan. Here's teichoic acid, and here's a lipoteichoic acid. Okay, so the difference between teichoic acid and lipoteichoic acid, this one is anchored in that lipid-rich cell membrane down here. Okay, so here's the plasma membrane or cell membrane, and there's a little periplasmic space in there, and then this is all considered the cell wall. All right, so let's contrast that with the gram-negative cell wall. As we said before, has only a thin peptidoglycan layer. Only approximately 10% of the cell wall by dry weight is going to be peptidoglycan, and after we gram stain it, they're going to stain red because they take up that last dye. They get decolorized really quickly when we add that alcohol to decolorize. It has a second membrane, so it has an outer membrane in addition to the plasma membrane. And that outer membrane, it's another phospholipid bilayer. There's proteins in there and something called lipopolysaccharide, which we often abbreviate LPS. Okay, and a portion of the LPS is lipid A. So basically, there's a couple of polysaccharides along with lipid A. And I went into a little bit more detail about lipid A in class because it was on my old lecture notes that I had to use. Mostly, we're going to talk about lipid A quite a bit in other portions of the course, but lipid A is also referred to as endotoxin. Okay, and lipid A is going to be released when the cell wall is disrupted. And guess what happens when the cell dies? Cell wall is disrupted. So typically it's going to be released in large amounts if you have a lot of gram-negative cells dying all at the same time. So if you have a patient with a gram-negative infection and you give that patient antibiotics and, lots of those cells die all at once, you have a massive release of endotoxin. And that can cause, when it's just, a few cells here or there dying, it's not really a big deal, but it can cause a lot of inflammation, again, if lots of cells are killed all at once. So typically a patient would have to be given some anti-inflammatory drugs to counteract the inflammation, the fever. The blood pressure can actually go down too low. Patient can go into shock if you have too much release of endotoxin. So we'll be talking about that more as we go, but it's kind of confusing. We thought that the toxin originally originated inside the cell. Now that we know it's actually part of the cell wall, it is that lipid A. Okay, so I have a picture that's going to show that a little bit more clearly. But here, let's label our cell wall of gram-negatives. Of course, here's the cell membrane. And here's the really tiny peptidoglycan layer. Okay, so this part here is all the outer membrane. It's got a phospholipid bilayer here, just like the cell, well, similar to the cell membrane. Oh, one more thing that I forgot to mention before here, this structure here is also unique to gram-negatives. This is called a porin. Okay, this is a non-specific protein channel used for transport. Okay, so this is the way cells allow things in or out of that outer membrane. And then these little structures sticking up here, this is the lipopolysaccharide. Okay. Let's see. Oh, this next structure, this next graphic is just another way to show that, but I like it because it shows the LPS a little bit better here. So the lipid A is this purple structure here that's embedded right in the membrane. So again, it's not causing any harm to the host as long as it's part of that outer membrane. But when the cell wall disrupts, again, that happens to some extent when a cell divides, but much more when the cell dies. That lipid A is released, and then will start causing problems for the host. Okay, so again, this shows the LPS a little bit better. I've dug up a few other graphics of cell walls. Sometimes when you see something a certain way, you remember it better. Again, here's a gram-positive up here. It shows that peptidoglycan is kind of purple squiggly lines. It has a little different way of showing the teichoic acid, the lipoteichoic acid. And then, of course, here's the gram-negative with the lipid A here and the polysaccharides that make up the LPS, those pores called porin proteins. Okay, thin layer of peptidoglycan here, but still has it. All right, so as we finish up here, there is actually one other kind of cell wall that is not really, that bacteria have, that are typically not considered either gram-positive or gram-negative, but it's considered its own special class of cell wall called an acid-fast cell wall. And I'll talk more about next time why we call it that. I didn't get to that in lecture today. But these cells actually do stain weakly gram-positive, so they don't, due to the structure of the cell wall, they don't take up those gram stains very readily. But they do stain a little bit purple, But again, they're really not typically considered either gram-positive or gram-negative. The main constituent of the cell wall, besides peptidoglycan, that makes it special is that it contains mycolic acid. And this is a long-chain fatty acid. It actually takes the cell a long time to synthesize this, and that's why they're typically very slow growing. But it also gives the cell wall kind of a waxy feel to it. And that gives the cell a little bit higher chemical resistance, a little bit more environmental stability. Okay, so basically because of that waxy cell wall, they will stay viable in the environment. Say it's a cell that somebody coughs out and it lands, on the desktop, that could stay viable for weeks on that desktop because it's not going to dry out due to that kind of waxy cell wall. We have gone to a lot of trouble to grow up these cells and to actually have a special staining procedure in order to see them, because there are a few organisms that are clinically really important, but it's mostly one genus that are considered acid-fast. There are a few genera that are considered partially active acid-fast, but the genus Mycobacterium is our main one that's considered acid-fast. And a couple infections caused by Mycobacterium, as you've heard of, leprosy, and the big one is tuberculosis. So I think I just heard a statistic, over 1.5 million people die every year, mostly in sub-Saharan Africa due to tuberculosis. So that's a huge scourge still. Okay, and next time I'm going to go through a little bit more carefully how we stain. So I'm going to start with this slide next time, but I did show this slide in class as sort of a summary. My labels didn't come out very good here, but it's just one more graphic comparing gram-positive bacteria with gram-negative with the Mycobacterium. And again, we talked about mycolic acid. We didn't really talk about any of this other stuff, so don't worry about that. There is still peptidoglycan there, even in the acid-fast. And the question that I posed in class here is, which kind of infection do you think is the hardest to treat with antibiotics? A gram-positive, a gram-negative bacteria, or a Mycobacterium? I'll go ahead and give you the answer to that. The Mycobacterium is definitely the hardest to treat. We talked about that waxy cell wall. Antibiotics, we don't have very many of them that are able to pass through that very hydrophobic cell wall. All right, so tuberculosis, really tough infection to treat. We have a few antibiotics that often work. But again, we don't have a huge arsenal of antibiotics that will pass through that. And then in terms of a gram-negative organism or a gram-positive, which is the hardest to treat with antibiotics? Well, you may think, oh, this has such a thick peptidoglycan layer that must be more of a barrier. But actually, it's not, because cell walls like this are actually pretty permeable to a lot of things. It's generally the cell membrane that's job to let certain substances in and certain other substances not. So actually, the gram-negative bacteria are much harder to treat than gram-positives. We have less antibiotics that are able to pass through this second outer membrane. So it's a little bit less permeable to antibiotics than what gram-positives are. So in general, and again, there's a lot of exceptions here, but a gram-positive, we have more antibiotics that work against gram-positives than we do against gram-negatives than we do, even against acid-fast. So we'll start next time with how to do that acid-fast stain, and then move on through our prokaryotic structures and get into the eukaryotic ones. Thanks for listening.